In laboratory settings, biologists have documented living organisms physically latching onto synthetic plastics and consuming them as fuel. This observed reality gained scientific prominence in 2011, following a landmark study by Jonathan Russell and a team of researchers at Yale. The team traveled to the Ecuadorian Amazon. By collecting stems from diverse woody plants, they isolated endophytic fungi, microorganisms that live within the plant tissues. Among the dozens of organisms they screened, one stood out, a fungus called Pestalodiopsis microspora. It demonstrated a robust ability to break down polyester polyurethane, or PUR. The evidence of this degradation is visible here. In these test tubes, the fungus was introduced to an opaque, milky suspension of liquid plastic. Over time, the fungus metabolized the polymer, turning the solution translucent. This chart highlights the most significant anomaly. The dark gray bars represent anaerobic conditions. Two specific strains were able to survive and thrive on plastic as their only food sources in environments completely devoid of oxygen. As these findings reached the mainstream, they fueled a popular misconception, the idea that we could simply dump spores onto our landfills and watch the plastic crisis disappear. While the biological capability is real, treating it as a magical, low-effort fix ignores the physical constraints of scaling laboratory science into the real world. Fungi have spent millions of years evolving into nature's primary recyclers. They possess a long evolutionary history of dismantling incredibly durable natural carbon structures, such as the lignin found in wood. They excrete specialized proteins called enzymes, shaped to bond with specific target polymers, severing their long chains into simpler nutrients. Russell's team confirmed Pestalodiopsis microspora produces a serine hydrolase enzyme capable of cleaving the synthetic ester bonds holding polyurethane together. This isn't an isolated case. Other common soil inhabitants, like Aspergillus tubangensis, have demonstrated similar enzymatic abilities when confronted with plastics like low-density polyethylene. Scientists have confirmed that specific fungi possess the innate chemical tools to dismantle synthetic polymers at a molecular level. However, many common plastics remain highly resistant to this process. For a fungus, not all synthetic materials are a viable food source. To see these limitations, we can look at research from the University of Sydney involving polypropylene, a tougher plastic used in everything from car headlights to upholstery. In their tests, the fungal cultures could not digest the raw polypropylene on their own. The chemical structure of the material was too dense and stable for the enzymes to gain a foothold. To trigger degradation, researchers had to artificially weaken the plastic first. They bombarded the polypropylene with ultraviolet light, high heat, and acidic solutions to oxidize the material before the fungi could begin their work. Biology is also fragile. Even with pre-treated plastic, these fungi require precise temperatures, moisture levels, and oxygen concentrations to survive and produce their enzymes. An organism that successfully dissolves plastic in a sterile, temperature-controlled incubator will likely starve or freeze if placed on that same plastic in a natural, uncontrolled environment. This friction is at the center of a major debate regarding missing plastic in our oceans. Some researchers speculate that marine fungi may already be degrading a portion of the millions of tons of waste flowing into the sea. At the University of Hawaii, researchers used experimental evolution to see if marine fungi could adapt. In just 3 months, they managed to increase the plastic feeding rates of certain strains by 15%. But the Journal of Fungi warns that real-world evidence remains thin. In natural settings, this process is incredibly slow and remains poorly understood. There is also a major gap in the data. Lab tests typically use pure crystallized polymers. In reality, the plastic debris in our environment is often degraded, dirty, and mixed with other materials that can inhibit biological growth. The sheer volume of the problem is overwhelming. Global production of new plastic now exceeds 300 million tons annually. While nature is adapting in real time, biological degradation simply cannot scale fast enough to compete with our current industrial output. This explains why we cannot rely on dropping fungal strains into local landfills to solve the waste crisis. In a landfill, lab-grown fungi are instantly outcompeted by wild microbes or killed by shifting weather conditions. A chaotic pile of mixed waste is not a viable habitat for high-performance bioremediation. The true promise of this field is more industrial than ecological. Instead of releasing the fungi, we can learn to harvest and mass-produce their specific enzymes. The future lies in engineered bioreactors, controlled facilities where these extracted enzymes are used to chemically dismantle sorted, hard-to-recycle plastics in a fast, efficient loop. But no biological tool can succeed without a massive reduction in the production of new plastic and a commitment to robust, conventional recycling infrastructure. Biology has demonstrated that synthetic materials are not chemically invincible. But for these cellular reactions to make a difference, we have to build the industrial infrastructure that allows them to function at scale.